o segundo dia do 4º Encontro de Biotecnologia e Bioprocessos. É um prazer estar aqui de novo, continuo com o meu resfriado. Ontem foi um dia de muito sucesso e de muitas palestras muito boas. Ontem foi um dia de muito sucesso e de muitas palestras muito boas. Gente, eu parei de falar porque eu fiquei me ouvindo uma segunda vez no YouTube, desculpa. Eu acho que pelo slide você está me vendo, você está vendo o Tassis, você está vendo o Sheila, você está vendo o Tony O'Donnell. Hoje eu vou falar do programação e eu vou fazer uma pequena introdução do professor Tony O'Donnell, que vai ser nosso primeiro palestrante. Mas antes disso, eu queria fazer aquele coisa da coordenação, de lembrar toda a nossa comunidade que antes do final do ano, por favor, atualize seus curriculares. Por favor, chorando, de joelhos, pedindo, rever seus curriculares, os professores também. Nós estamos no último mês do quadriênio e nosso trabalho está avaliado do que nós temos como comprovar no Curriculares. Curriculares faz parte do sistema do Sucupira, que é o software da CAPES que a gente usa para avaliar nossos programas. Então, isso é uma lembrança de todos os professores e todos os alunos. E, muitas vezes, eu sou um que não fica tão atualizado. Então, essa é uma mensagem para mim também. Então, estamos no último mês de Curriculares, vamos fazer isso. Ontem, só lembrando, a gente conversou sobre quais são as perspectivas para a biotecnologia. A professora Andy Whiteley falou um pouco sobre o monitoramento de vírus lá e as lições aprendidas e a necessidade com países enormes como a Austrália, como o Brasil e alguns dos outros países que geograficamente são enormes, os desafios. Depois a gente ouvimos todas as histórias. Depois a palestra do Márcio sobre cetários viridis, nosso planta modelo, e o resto. Hoje, Tony vai falar, depois vocês vão falar, e eu vou falar um pouco sobre o UFRJ PR2, onde o programa encaixa nos programas de pós-graduação. Eu vou falar sobre PRINT, que é o programa de pro-internacionalização da UFRJ e como o PBV encaixa dentro dessa pro-internacionalização e por que ser internacional é importante para o nosso programa e importante para a nossa avaliação com o programa nível 6. E como nós, todos os professores, podem ser mais internacionais, certo? E no final do dia, nós vamos ter uma palestra sobre inovação. Então, isso vai ser como nós fechamos. Gente, são 9h05. Por enquanto, só tem 26 pessoas inscritas, então vou esperar um pouquinho mais, porque eu acho que as pessoas estão esperando, talvez, às 9h30, para ver o professor Tony. Tony, enquanto eles estão chegando, eu vou falar um pouco sobre o professor Tony. O professor Tony O'Donnell foi meu orientador do meu doutorado, muitos tempos atrás. Eu fiz meu doutorado entre os anos 94 e 98. Defendi minha tese em 98. Estudei a microbiologia do solo através do genes 16S. E o título da minha tese era Ecologia Risosférico usando o gene 16S. Então, para vocês que são da área de microbiologia, é verdade. A gente estava fazendo isso em 98, 22 anos atrás. Então, desde então, a gente aprendeu muito sobre biologia molecular. O professor Tony sempre foi um cientista do solo, sempre foi na frente de todas as tecnologias. Então, ele, na carreira dele, estudou provavelmente todas as metodologias e sempre estudou eles um pouco antes do que a grande maioria. Então, enquanto ele era meu orientador na Universidade de Newcastle, ele era o decano associado da área de pesquisa. Sempre foi atuante na área de pesquisa. Depois disso, ele foi o diretor do Centro de Pesquisa em Sustentabilidade da Universidade de Newcastle. Ficou nesse cargo por vários anos já, professor titular. depois mudou para a Universidade de Western Australia, em Perth. A Universidade de Western Australia, em Perth, é parceiro da UFRJ, mas principalmente é uma das universidades bem parceira do nosso programa de pós. Vocês talvez não sabem, mas eu acredito que, se não todos, quase todos os professores do nosso programa têm parcerias e têm contatos com universidades internacionais. E é o objetivo do programa que nossas pesquisas tenham impacto internacional. Uma das melhores maneiras de ter impacto nacional é estudar com parceiros internacionais. Até, se tiver a oportunidade de estudar fora, aproveita. Se alguém te convidar, tenta. Se existe prêmios para viajar, ou uma possibilidade através de consulados, tem uma chance de estudar fora, pega essas chances. Por quê? Você vai expandir seus horizontes, com certeza. Você volta uma pessoa um pouco diferente. Você volta com uma visão mais global, mas global no melhor sentido, no enriquecimento de cultura e de conhecimento. Não global no sentido de tudo igual. Eu não estou falando a gente viaja para ver um McDonald's, não. A gente viaja para criar novas ideias, entender culturas novas, ganhar novo conhecimento e para depois compartilhar isso. Então, o Tony sempre foi um orientador que aceitava alunos internacionais, sempre teve essa visão internacional. Eu acredito que a visão internacional que eu também tenho, eu ganhei do meu orientador Tony. Então, outra coisa que eu acho interessante é manter os elos, né? Você cria elos em outros países, cria elos com outros pesquisadores, e isso é importante, porque o impacto depende um pouco do nosso pensamento, o impacto depende um pouco de como a gente compartilha as informações e como eles são divulgados. Então, nosso evento, que está sendo gravado e compartilhado, vai ter um impacto além do que um evento que não foi gravado, que não foi compartilhado. Eu entrei no site no canal do nosso programa de pós hoje de manhã, e eu fiquei feliz e surpreso com o número de visualizações do que foi feito ontem. Ontem, a gente teve, na média, 800 pessoas participando do evento. Mas eu já vi que as visualizações já estão em mais de 100 em um dos vídeos, em 200. Então, os trabalhos de vocês, nossos trabalhos, estão tendo um alcance maior. E quando a gente trabalha em parceria internacional, esse alcance fica maior ainda, ok? Então, o Tony foi para a Austrália. Em 2008, teve a reunião do ISME, que ficou em Cairns, no nordeste da Austrália. Eu fui nessa reunião, a minha aluna, Ida Carolina, que agora é professora no ESO, nós fomos para a Austrália para apresentar nossos trabalhos, e naquela época eu acho que eu fui visitar Tony, e conheci a University of Western Australia pela primeira vez. Eu voltei, eu não sei, uns 4 anos depois, e eu voltei também em 2015. Mas desde então, a professora Bianca já estudou lá, a professora Fernanda Reinert já estudou lá, a Ana já estudou lá. Então, a gente tem já um intercâmbio interessante entre essa universidade. E não é só UWA, mas hoje eu estou falando de Tony, porque ele vai dar uma palestra para a gente. Mas, se você pensa nos seus grupos, se você não tem no seu grupo, com seu orientador, uma parceria internacional, é isso que nós queremos. Mais tarde, hoje, eu vou falar que, além de parcerias internacionais, a gente quer parcerias industriais. Então, todos de vocês e todos os professores têm que ter, no mínimo, uma parceria industrial, uma parceria internacional. Isso é o mínimo. Muitos já têm isso, certo? Então, eu estou falando, os números já estão melhorando, estamos em 34 pessoas, já falei 10 minutos. Eu não sei para quanto tempo Tony vai falar, eu convidei ele para falar 20 minutos. Então, é isso. Então, eu acho que devo falar para mais 20 minutos até 9h30, né, gente? Então, além dessas parcerias, além disso, Eu queria, então, vamos aproveitar esse tempo para falar sobre a nossa avaliação e vamos falar sobre a avaliação do programa e seu papel. A melhor coisa que vocês podem fazer é ter seu curricular atualizado dos seus orientadores. Ver que está tudo lá. Uma coisa que mudou nas avaliações de programas na área de biotecnologia são o que é um produto, né? Vocês foram sempre treinados para valorizar artigos publicados em revistas indexados com impacto. E, sem dúvida, isso é importante. Mas todo mundo aqui também cria produtos biotecnológicos. Você cria protocolos, você cria conhecimento que nem sempre cabe num artigo científico. Isso não quer dizer que não tem valor. Então, se você cria novos protocolos, eu queria que você registre isso como um produto técnico. Coloque ele de uma forma formal, crie um código para ele e valorize isso nos seus curriculares. A gente tem como valorizar isso. Eu queria também que, onde tiver parcerias com empresas, que isso fique registrado. Ok? Então, gente, eu acho que eu estou enrolando mais que suficiente. Estamos em 34 pessoas. Agora eu vou passar a palavra para Tony. Eu vou falar inglês um pouquinho e passo para ele. Okay. Tony, I'm now going to speak a little bit in English. I'd just like to thank you very much for agreeing to be a part of our end-of-year postgraduate program meeting. This is our fourth meeting, so this is the fourth year where we have held a conference at the end of the year to celebrate another year of work, to allow the students to show their production, show what they've been doing, for them to all speak in English, and to, in 5 minutes, give a sort of a flash talk of what they've done in the last year, or if they've arrived more recently, what they've done since they've arrived. I think I learned from you that it's important to stand up and talk about your work. I think that was one of your lessons, and what we've learned by insisting that our students do the same is that they benefit from it. So for the last when we started this 4 years ago, it was an awful shock for most of our students to have to stand up and speak in English. They wouldn't have had a problem speaking in Portuguese, but making them speak in English was a tough call for more than half of them. Now I would say that 2/3 of our students are quite comfortable speaking in English, and the other third speak in English, and they're suffering, but they do it. And I think that's a really good thing. I'm not sure, I know it's the middle of the night where you are, so everybody, where Tony is, Tony's in Perth in Western Australia. It's 8 o'clock in the evening. It's very hot where he is. There's going to be a thunderstorm soon, and we're worried about the internet, so hopefully that won't work. Tony is a professor of soil science. He's a leading thinker. He's a highly cited soil scientist, and he's going to share his vision and his knowledge of what's going on and what's important and what drives changes and interactions in the soil. We're going to go into into the microbial scale. We're going to go through the eye of a needle and have a look at what's happening in soil. So Tony, thank you very much. Tony is one of our visiting professors, and if you want to exchange emails with him or have more information about what he's doing, then afterwards get in contact with me and we'll make that link. Tony, thank you very much. It's you're on.
Okay, Andrew. I need to share my screen?
Yes, yeah.
So can everyone see that? Is that coming through?
Yeah, that's coming through now.
Okay. Can I just start by thanking everyone? And I know it's early in the morning for you, and I wish I was actually in Brazil. I had actually planned, as Andrew knows, to be across, but I think COVID's puts paid to a lot of our international traveling. So I'm very, very happy to do this. And what I want to do is to talk a little bit, as Andrew said, about the importance of microorganisms in soil, and in particular, what do we actually know about those organisms, and how can we actually start to start thinking about them as something that we can use and we can manipulate. I chose the title Through the Eye of a Needle because I think it epitomizes exactly what the big challenges, the big intellectual challenges in actually studying microorganisms in soil. And the study of microbes in soil has gone through a number of different cycles. And I want to touch on a little bit about some of those cycles. I'm actually going to draw on the work of a lot of students over the years. I've been fortunate and been blessed, Andrew was one of them, in having a good group of PhD students that have done a lot of this work. So I want to start by acknowledging them. I don't quite know what Andrew said about me at the beginning, because despite having traveled to Brazil regularly for the last 30 years, I do not speak any Portuguese. But I started my career in Glasgow, and I trained initially as a medical bacteriologist, and then moved to Food Science Institute, where I worked on pattern recognition and essentially multivariate statistics. And then I moved back to Newcastle, moved up to Newcastle, and joined an organic chemistry department. And I worked as an organic chemist for 5 years, and then joined the agriculture faculty as a soil scientist, and then retrained after about 10 years as a molecular biologist. I only tell you that because some of you are probably starting out on your career wondering what you're going to do. Well, I've changed my discipline, changed my area so often, and to be honest, I don't regret any of it. So what I'd like to do is to take you through the presentation, and as Andrew has said, I'm more than happy to take questions at the end. One of the reasons that we really want to study soils and soil microorganisms is that land and agriculture, food production, is actually so important to sustain life on Earth and to maintain the social structures that we all have. In order for soils to be functional, they need to deliver ecosystem services. And those ecosystem services are more than just food production. They also store water. They purify water. They're a repository for pollutants. We use them to dispose of wastes where they get biodegraded. So having productive and functional soils is really very important. The microorganisms are key to these processes. And on the right-hand side, we've got some of the founding fathers of soil science and soil microbiology. Dokuchayev, a Russian, actually was the first person to describe soils as a system, and largely forgotten, I think, until relatively recently, that it's actually very hard to study components of the soil in isolation. It all interacts so closely. Physics, the chemistry, the biochemistry, the biophysics, all interact to actually give you a functional soil. I mean, part of that departure away from system science was some of the brilliant work done by people like Winogradsky, the guy below. Winogradsky was the person who started doing microcosm studies. So we went from sort of studying soils in the field to taking samples and trying to reconstruct microcosms, the famous Winogradsky columns some of you may well have used, that he used to study microbial processes in soils. And then Beijering will be well known in Brazil, nitrogen fixation. A lot of this saw a focus on single organisms. And when I started in this field, a lot of the work I did was around isolation of single organisms, screening for novel products, supplying biotech companies, identifying novelty, so identifying areas that had organisms that had never been screened before. But we really needed to get back to some of Dokuchayev's original ideas about thinking about soil as an interconnected system, something that connects across different scales and across different components of the soil system. And this is where the idea of the soil microbial biomass first became popular. In 1966, this man's David Jenkinson. I knew him very well. I worked with him back in the early days. He developed things like the Rothamsted soil carbon model for predicting carbon turnover in soil. And David was a chemist by training, took a different view of the microbial biomass in soil, the biological activity in soil. And David really developed this system, this idea of the soil microbial biomass. Nowadays, we probably call it soil microbiome. And he developed a suite of methods for actually measuring the size of that biomass. And the people who actually followed David's thinking were really very sure that the size of the microbial biomass was more important than its composition. And I did a lot of work in the early days looking at things like developing methodologies for measuring biomass sulfur, biomass phosphorus, so we could actually measure the transit of nutrients, key nutrients, through the microbial biomass. The importance of this very, very limited amount of carbon, usually less than 1% of total soil carbon, is that almost every transformation process that occurs with carbon or even with inorganic materials goes through this 1%, this soil microbial biomass. And that's why it's often referred to as the eye of the needle. Everything has to go through that very small percentage of biomass, of carbon in the soil that represents the soil microbial biomass. And Kevin Tate produced this book in the, I can't remember when it was now, but that actually refers to this, makes a point of the eye, shows the eye of the needle. Everything that actually we eat, we do, we go, all that transformation processes goes through that microbial biomass. So effectively what the microbial biomass is doing is it's aggregating this 1 gram of soil contains between 10 to the 7 and 10 to the 10 bacteria, and probably also thousands of taxa, different taxonomic groups, phylotypes, genotypes, etc. So lots of diversity in there, but David's argument was the size of the microbial biomass was the key thing. So one of the things that's intrigued me for many, many years is, what is that assumption correct? Is the composition of the microbial biomass of limited importance in terms of soil function, or is it just the size that's important? And studying microbial biomass is actually quite difficult, especially if you want to understand what's affecting its composition. So what is its structural composition? What's it made up of? What kinds of organisms? Is it fungi? Is it bacteria? What kind of bacteria? What kind of fungi? Because there are so many other things that interact and impact on that soil microbiome, the soil biodiversity management. So farmers are actually farming microbial diversity when they put in crops. The plant interacts with the microorganisms and will change its diversity. The management affects soil physics and soil chemistry, which again interact with the microbial biomass and change its composition. Then you have the activity of decomposer organisms that are not bacteria, like protozoans. You also have larger animals like ecosystem engineers, the soil animals, which things like earthworms and springtails, mites, things like this in the soil that actually mix all of this stuff up. So it's a very, very complex system. But we do know that we can actually change the biota. We can change the structure of the microbial biomass. We can also change its size. And I was very lucky to work in Newcastle on an experiment, a long-term experiment called the Pals-Lees fertilizer trials. This experiment was set up in 1897, and we don't have enough long-term experiments around the world, but since 1897, this site, these plots have been treated in the same way. They've received the same amount of organic and inorganic manures since 1897. This has resulted in a very stable microbiota within the same local environment, but where the populations have actually changed. And we can see that they've changed. So you can see down here, there are 13 different plots. pH has been altered by the management. So the chemistry of the soil has been changed. This is the amount of the hay that we're getting off. This is a hay meadow trial. The amount of hay we're getting off the land, and then the different timing of fertilizers, and also the amounts of nitrogen, phosphorus, and potassium actually added. It was quite a comprehensive trial. We know that it's affecting the biomass. These are some measures of biomass carbon. So this is using Jenkinson's methods to get an idea of the total size of the microbial biomass in the soil. And you can see here that in some of these plots, the ones in blue, these are all plots which have either had no inorganic fertilizer added or have had organic fertilizer added. So less impact on the biomass, but plots 7 and 11 receive quite high amounts of ammonium sulfate as fertilizer, so since 1897. So the nitrogen that was supplied as ammonium sulfate. And you can see it's reduced the microbial biomass carbon. And if you walk down these plots, you can see it's affected the vegetation. So we can actually change the size of the microbial biomass and the impacts of that microbial biomass through fertilizer treatment. We can also see that on these plots that other properties have also changed. The water infiltration rate, so when water falls, how fast does it move into the soil, has been changed. And these changes are correlated with the size of the fungal biomass as measured using a glycoprotein glomalin or a fungal membrane sterol ergosterol. You can see that where the value of these are high, the water repellency is high also. So we can see that the management affects the microbial biomass in soil structure, function, and that then has an measurable impact on soil properties. And where I live at the moment, in Western Australia, we've been working on this area with BASF. We have very, very hydrophobic soils, water repellent soils. So in Western Australia, it's dry and drying, the climate, but very often the water lands in the soil and runs off. So we've been looking at ways in which we can develop polymers, change the microflora that would allow water to infiltrate. So this is actually a very important property of soils, is water repellency. And you do have water repellent soils in Brazil as well. If you can also look at this in other ways. You can look at, I did a lot, as I said, I spent some time in an organic chemistry department after I finished my PhD. So we developed methods for analyzing membrane lipids. So looking at the structure of bacterial cell walls. Now these bacterial cell walls, the structures change according to the taxonomic grouping, but you can use it as a biomolecule, a biosignature, to look at just what's happening within that black box, is the soil microbiome, the soil microbial biomass, what's actually happening. And this shows you that in these plots here, this is plot 711, these are the ones that receive nitrogen as ammonium sulfate as a fertilizer. We get quite different, quite distinct microbial communities developing. Probably are likely as a result of the fact that ammonium sulfate causes acidification of the soils, because all of these soils, 711, all have lower pHs, about 3.8 to 4. If you take this out, take these analyses and just analyze this separately, you can also then see that there's a phosphorus effect. So the microorganisms in these soils were getting inorganic phosphorus as well. And you can see that there's a difference between the amount of organic matter, soils treated with organic matter and soils treated with phosphorus. So we can actually change this microbial biomass. We can alter the contents of the black box, but we still don't have any idea from these methodologies what's actually happening, what is actually changing. We know that we're affecting change, that those changes are actually having an impact on soil properties, but what is it we're actually changing? So in order to do that, we had to develop and use some of the molecular methods, more molecular methods. And I've spent the last 10 or 20 years now really just looking at trying to sort of optimize those methods and try to use them to give us more insight into what's actually happening at individual organism, individual phylotype level. So the question we're really looking here was, are specific taxa responding to management? So this is the data from the Pallas-Lees data. So this is 16S rRNA data. And what we've done here is just do some very simple rarefaction curves, looking at the alpha diversity and plotted them out. And immediately you can see that the different plots have a different alpha diversity. This is some we've revisited this data. This is some relatively recent work, a different way of looking at these numbers. And that the interesting thing, plot 6 is marked here. This is the control plot. So this is a plot that did not receive any fertilizer. So this is the control plot that had received neither inorganic fertilizer nor organic fertilizer. And one of the questions that we were asked to look at a couple of years ago was whether or not organically managed soils are more biodiverse than inorganically managed soils. And it was actually quite a challenge at the time. But what this shows us that all of these soils here, so all of these plots, this one here would plot 2, plot 3, plot 5, all of these above this are all organically managed. So they all have organic matter added to them in some form or another within the management regime. I'll come to that in a moment. And with the exception of this one here, plot 4, all of the ones below the line actually have farmyard manure, inorganic fertilizer added. This one, plot 4, has organic fertilizer added, but very, very little. So it's not had much of an impact on the diversity. But what this actually shows, I think, very clearly is that the addition of organic manure actually increases microbial diversity in soils. We can also look at this in a slightly different way. We can actually start to think, we can cluster these not as individual plots, but as treatments. So we can actually start to look at the treatment effect to actually try and unpick that relationship between an increase in diversity due to the addition of farmyard manure, organic matter, and a decrease due to inorganic amendments. So you can see here again the control soil. And this is also an interesting finding. Yes, organic manure addition actually increases the overall alpha diversity in these soils, but it's actually less than we get when we add a mixture of organic and inorganic fertilizer. So when we add manure with inorganic nitrogen or inorganic phosphorus, we get it has a bigger, more profound impact on the alpha diversity of the soils. Whereas, again, all of the inorganically treated soils have fallen below the control. So the effect of inorganic fertilizers on their own has been to reduce the alpha diversity in those soils. The fertilizers are also added in different ways according to a management regime. So some plots are actually fertilized every year, either with organic or inorganic or a combination. Some get fertilized every 4 years, some get fertilized every 2 years, and the control plot 6 has never had any fertilizer added to it. Again, an interesting finding, because some of our data suggested that there were differences between, at least in the fatty acid profiles that we looked at, between plots where the treatment was being applied every year. So if we want to optimize diversity, the addition of fertilizer every year trumps over adding it every 2 years or every 4 years. So using this really very, very simple approach, we can start to answer some of these key questions about fertilizer addition, whether or not organic or inorganic is best. We're starting to be able to answer some of those questions. What those plots don't actually do is actually show, again, tell us what's actually changing. So what we're seeing here is big changes in diversity, but we can actually drill down and start to ask the question, well, what is changing? So we've also been doing some work looking at what these sequences actually key out as. And one of the interesting things I think it's emerging over the last couple of years is that a very useful paper, the Global Atlas of Soil Diversity, was published a couple of years ago. And that paper showed that whilst there's a lot of diversity in soils, over many, many different soil types from across the world, only 2% of the total available phylotypes were found in 55-60% of the soils. Now we've got the similar thing here. Here you can see that somewhere between 55 and 75% of the organisms, the phylotypes, the sequences we're pulling out of these soils belong to 4 taxa: the delta proteobacteria, bacilli, acidobacteria, and the actinobacteria. Now those actually are the same groupings as the World Atlas found with the most common groupings of microorganisms and of microbial phylotypes in soils. So these results are supporting what we might actually expect. Also, these are on the same parent material, and I'm doing also interested in finding what relationship there is to the parent material. You know, are soils predetermined to have certain types of organisms, certain types of taxa because of the parent material on which they form? But you can see here very, very clear that some of these groups, the delta proteobacteria, the bacilli, and the acidobacteria and the actinobacteria, and Andrew will know that we've worked on all of those at various times over the last 20 or 30 years, actually dominate these soils. Interesting thing, delta proteobacteria is where these sulfate-reducing bacteria normally come. And when I saw this result, I was a little bit surprised that there were so many in these soils. But then let's not forget that the SRBs actually use sulfate as their terminal electron acceptor. And these soils have been treated large numbers with sulfur, ammonium sulfate, for many, 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 many years. So maybe it's not surprising that we've got a population of organisms that's adapted to using sulfate as a terminal electron acceptor. So one of the things that often asked about microbial diversity is, does it affect function? And this set of results, when I saw them, this is using MGRAS to look at the functional diversity. And this really this really surprised me. We've got that the different sizes of biomass. We've got differences in the 16S phylotype distribution. But when I put this graph together, I thought I had put the same figure on 4 times. So this is basically a very simple, arguably quite a crude way of looking at what the impact of fertilization over more than 120 years, 123 odd years, has actually had on the function in these systems. And this is by looking at sort of the distribution of functional genes, as a selection of functional genes. And you can see it's actually very little. So the functional capacity is still there. So whilst we might have changed the structure, the functional capacity, the ability to deliver these key subfunctions hasn't actually changed. So we really need to do a little bit more work on this, just to try and work out what's actually going on here. Maybe not too surprising. Similar types of soil, the microbial populations are changing, but the core functions of this soil are remaining. If you compare these with other soils from different sites, you get different distributions in these patterns. But all of these plots, these 4 plots that we looked at with metagenomic analysis, we found that the actual functional capacity, the functions that were present were very similar. So what are we now doing? We're actually, at the moment, we're working on a proposal to try and actually tie a lot of this together. And some key facts I think that are intriguing for soil scientists is that if you take 1 gram of soil, represented here by that tiny little cube, it has about 10,000 protozoans, 10 to the 7 bacteria at the lower end, and 5 kilometers of fungal hyphae. And you can use some sort of ballpark figures and work out just how much in terms of volume that would be, or surface that would be. And this is work that John Crawford and Ian Young did a couple of years ago. Only 5.5 times 10 to the minus 4% of the surface area is actually covered. So most of that 1 gram of soil, that 1 centimeter cube of soil, is actually space. Yet that is responsible for all the transformations. That is the eye of the needle. That's where it resides, but most of it is space, voids that are filled with water or periodically filled with water. So the real challenge for us is to actually try and go from what's actually happening at these surfaces, so redox changes on surfaces, absorption, desorption reactions on surfaces. We can study at the molecular level, what's happening to the microbial population. We can use isotope techniques and visualization techniques to actually see where these things are located on surfaces. How do they affect aggregate formation? Very, very important in agriculture, gives the soil structure, allows penetration of water. How does it affect this? But then we have this big gap. How do we go from here up to a horizon, to a field, up to a catchment area? This is what we're now being asked to deliver for government. They want to know what this is. The scientists are all interested in what's going on down here, but they want to know what's going on up here. So we're pulling together a big program to try and look at these questions of scale. So what's actually linking these different scales together? Some early work that Ian Young did showed that these relationships actually break down after a few millimeters. So it's a really big challenge. Water is obviously very important because water will fill these pores and it will allow organisms and nutrients to move around. So what impact does this have in a drying climate? So we've been doing, I mentioned before that we are actually also been looking at how the parent material affects these microbial functions and also microbial structure. And this, I put these in here just in case anyone's interested in reading a little bit further. But it's interesting living in Western Australia. Western Australian soils are very old, and particularly on the west coast of the continent, these are very, very old, ancient soils, geologically ancient soils. Those of you who have studied processes like nitrification in soils will probably have read many, many times that the ammonia oxidizing archaea are the predominant organisms responsible for nitrification in soils, and that the ammonia oxidizing bacteria play a secondary role. Well, in Western Australia, the situation's reversed. Large parts of the state, you cannot find ammonia oxidizing bacteria in any great number. Ammonia oxidizing archaea in any great numbers, and the systems are dominated by ammonia oxidizing archaea. We're interested in knowing why this is the case, but it turns out it's to do with copper. The soils are so highly weathered in Western Australia that the copper that is present on the eastern side of the continent has been depleted, and the soils are rich in iron. And one of our postdocs had a bit of a brainwave and realized that, Sasha Jenkins, that actually the ammonia oxidizing archaea will tend to use copper as a cofactor in ammonia oxidation, and the ammonia oxidizing bacteria will tend to utilize iron. So here's a direct link between the underlying parent material and a functional group of microorganisms. So one of the things I'm interested in is to what extent can we actually start to understand this better, develop better models, develop better management practices, so that we can actually manage this. For example, there'd be no point in putting certain types of fertilizers onto some of these soils because they will not be transformed. We've also been looking at how populations differ down depth, and again, this idea of the parent material and how populations change or become more similar the closer you get to the parent material. And again, because of the microbial activities and the effects of weathering on the release of chemicals from the parent material, like manganese, iron, potassium, nitrogen, etc. So those are some studies we're doing. We can also see at a higher scale changes, and I put these two slides in. This is an analysis that was done of the size of the actinobacterial and fungal communities in about 300 soils from across Australia, from the east and the west. And again, you can see that the soils in the west are quite distinct, the black triangles, from the soils in the eastern part of the continent. And this is with actinobacteria. Now, this is not zero. This scale needs to be redrawn to a different scale, but much lower levels of fungi in the West Australian soils than in the soils from the eastern states, from Queensland, Victoria, and etc. And also actinobacterial numbers tend to be higher in Western Australian soils. Now, we're interested in knowing whether or not these organisms, actinobacteria and the fungi, both have a similar growth habit, and whether or not, as with the ammonia oxidizing archaea being replaced by ammonia oxidizing bacteria, the actinobacteria performing a similar role in the West Australian soils as the fungi are performing over in the eastern states. So lots of really interesting things to explore. I love working in soils. It's been a challenge, an intellectual challenge. Nothing is straightforward, and I actually think that things are starting to come full circle. We're moving back towards a situation whereby we're looking at the activities of individual organisms and the importance of spatial scale and distribution as opposed to microcosms in one or two samples is becoming increasingly important. The work that I've got going currently is a large scale regional analysis, working with a pedologist, somebody who studies parent material and soil structure, looking at a transect across New South Wales to look at how the microbial diversity changes. And this is in keeping with the problems that Australia is going to have with climate change and drying climates. So I'll leave it there. I'm very happy to take, it's a bit of a gallop through everything, but I'm very happy to take questions. And I'll hand back to Andrew.
Okay. Thanks, Tony. Thank you for a very interesting talk. I was I'm now back in the studio with you in this other webpage, but out there on YouTube, there's a list of questions being put out there. A couple of things that struck me in your talk, Tony, were well, a few things I thought was very interesting, and I'm just going to reinforce that, the importance of long-term experiments. So the Palliser experiments that you showed, I don't think our students have ever seen a 100-year experiment. And I think the way PhD students are trained, all of us are trained in academia, is that we're expected to have results. I think once upon a time, probably in 3 or 4 years in a PhD, and maybe when I did my PhD, that was okay to have some kind of result, something interesting to say after 3 or 4 years. But now we sped that up, and we want people to find interesting little bits of information that are publishable within a year. And sometimes the big questions just can't be answered on those timescales. So I just thought that was extremely valuable to share, that there are some 100-year experiments, and some of the data from that. But I've obviously seen that data from that experiment 20 years ago, and it's very nice to see it updated with the molecular data. But I was a bit like you when you showed me the graph of the metagenome analysis and those 4 pie charts. I looked very hard at that slide, and they are very similar. I was thinking, and so immediately, being the skeptic that I am, I was thinking, well, does this method actually measure something? Is it measuring what's going on? It's obviously telling us which genes were detected and their relative abundance. It struck me as strange. I thought, is this valuable? Is that really a good measure of function? Because I noticed that there's a significant pH shift between some of those slots. Some of them were, I think one was pH 3.7, and the other one was I think there's a couple of it 5.5.
5.8, yeah, 5.8 to 3.8, 2 pH.
So 100 times more acidic. And that must have an impact, but it doesn't seem to. What do you think? I mean, what do you think that the basic gene functions are always going to be there? So it's a good, or isn't? Is it a good measure? That was one question I had for you.
I mean, I did say I think we need to do that again. I was not convinced by either. Or I mean, it may well be that the resolution's just not good enough. We put in a metagenome and let the analysis just tell us what's there. It may well not be good enough. If we make process measurements, and we have done those as well on these soils, we get differences in process measurements. So the soils actually produce more or less CO2, depending on the size of the microbial biomass and its activity. So other measures have used techniques, again, also challenging, but the microbial respiration, things like dialogue and stuff, you'll get different patterns with those as well. So I think we have to be very careful with taking that metagenome data at its face value.
Yeah, and I think I've raised that just because all of our students are, all of this generation of students are students that are studying genomic approaches to everything, and that's necessary and good, obviously. But sometimes I think stepping back and looking at some of the other methods, I think that it's going back is that we really need 2 or 3 methods at least to support a hypothesis. And I think the danger is that a lot of us, including myself, is that we're focused on molecular methods and very often try and reach conclusions that are maybe not safe, I would say. The other thing I thought was really interesting is, and I wasn't aware of, was the possibility of differences between archaea and bacteria based on the levels of copper or iron, or the lack or
that's the only thing we could tie it down to. That's our hypothesis. There's a lack of copper in the soils in Western Australia has meant that AOBs have dominated those soils.
Yeah, no, and I think Western Australia has got all sorts of very curious, interesting things. You've got the oldest rocks on the planet. You've got the oldest soils on the planet. So for some of our students, if you get the opportunity to study in Western Australia, you should take it, especially those in the more agricultural soils side. In fact, any side. We're talking about rocks that are 3.5 billion years old. That it's the oldest stuff that we've got on the planet. So it's an amazing place. The other thing that struck me is that you've been doing some recent studies comparing Eastern Australia with Western Australia. For people, the majority of our students don't know Australia, haven't had the chance to go there. Australia is a continental. It's about the size of Brazil. The middle of it is a big desert. I'm not sure if I'm simplifying too much. Is most of it a desert? Does it rain as much on the east and west? How does it work?
All those samples are actually, that work was funded by the grains industry, the GRDC, Grains Research and Development Corporation. So all those samples were from the bits around the edge where we can actually grow wheat. So all of those are the wheat.
And yesterday in some of the talks, the subject of water, the importance of water reappeared. And I also thought it was very interesting. I doubt as well that the majority of our students have thought about or had so well explained what does 1 gram of soil look like, and the fact that it's it's a sponge, it's a matrix, it's gaps, it's spaces, and that sometimes there's water in them after it's rained, sometimes there isn't, and the connectivity of it is, and the challenges of scale up. So I thought that was really valuable too. I was curious about pH. Is there a general pH pattern in soils, or all the soils in Australia acidic because they're old, and how does that work?
No, they're not all acidic, but I think pH is well established as a major driver. There's been studies done in transects done through North America and many places where pH has been highly correlated with changes. But then pH affects so much in soils. It's not directly on the organisms. They affect cation exchange. They'll affect water availability. They'll affect the availability of nutrients, exchange of surfaces. So pH it has such a disturbing effect on it, on an overarching effect on everything.
Yeah.
And it's actually difficult with these systems to unpick pH from other treatments because it is
it's also surprisingly difficult to measure, although
Yep.
it appears in papers all the time. I remember having fun trying to measure pH in soils back in Newcastle. Tarcísio, tem algumas outras perguntas aí? Você achou?
Yeah, for more question right in the end. I will post them at the bottom of the screen.
Okay.
First one is from one of our students. Is it possible to combine a genomic study with a soil sequential extraction process?
I think so, and I actually, it's a really good question. I think it's a sort of, I was going to add this with when Andrew was talking. We really shouldn't just be relying on genomic studies. I think when this all started out, you had trouble getting genomic studies published on their own. You had to have process measurements. So maybe if Claudio can explain what he means by a soil sequential extraction process. Is that to take out different types of elements each time and see what happens?
Well, I think so.
I wasn't quite. So maybe you can email me. I'm more than happy to answer an email if he sends me an email. But
Yes.
I think the idea of combining some sort of process measurement or what's actually happening. We've actually done work where we've looked at trying to change pH and how over a 6-10 week period, how we've also tried to shift the stoichiometry of the soil. They all revert after a set period of time. When you take the stressor or the treatment away, they all revert to what they were before. And that's really quite interesting as well. They're quite resilient to change, resistant to change.
Yeah, okay. Thanks for your answer. One question from one of our professors. The microbe population in tropical regions is quite dynamic because of the high temperature. How the use of organic or chemical fertilizer can affect this microbe population?
I haven't done a lot of work in the tropics. I think it would be very similar to what we're seeing in temperate soils in the northern hemisphere, but also we're seeing similar things in Western Australia, which goes from a Mediterranean climate in the southwest up to a tropical climate in the north. We're seeing similar things. I think you would see, depending on how much organic matter is going in, I think you would see similar changes, but we're not. You'll see I remember one time giving a presentation where I said a throwaway line. I said that microbial populations in soil change if you walk across them, and a PhD student put up their hand and said, "No, they don't. I've measured it." The fact that somebody went out and measured that really surprised me. But he said, "No, they don't change." But they do. They are very dynamic, as Jean-Louis has said. They are actually very dynamic.
Okay, thanks. One last question from one of our students. New technologies aimed at agriculture accelerate processes and impose on nature a kind of natural grain selection. Can this long-term process change nature's production matrix?
Is that question 2 or 2 new technologies?
Well, can you help me there?
Now, I mean, I think the new technologies are, going to change agriculture. The big thing in agriculture at the moment is innovation, and we're looking at a whole range of technologies that we can apply to agriculture, like many other people. Natural grain selection. I'm not sure what's meant.
I think I'm not.
Just rather than plant breeding or traditional breeding or genetic engineering.
I think that's the question. Is it just a normal natural evolution and how we can impose biotechnology for improving grain production? I think that's what she's saying. I think that's a plant genetics question, really.
Well, we're also looking at, one of the things that we've been looking at is using plant genetics to actually change the exudation profiles from roots and actually change the rhizosphere so that the rhizosphere becomes more able to trap, uptake nutrients. We're also looking at development of polymers that we can put into soils so that they will actually hold more water, or better delivery of inoculants. That's what we're doing with the industry. Better delivery of inoculants to roots, and sort of trying to engineer the rhizosphere of plants, food crop plants.
One more question. Is that time? Is that right?
No problem.
How the Anthropocene impact the study of soils? Would destruction be an obstacle for future studies?
Yes. Simple answer. Yes. I think we have to be we all need to be very careful that we are losing soils very, very quickly. And, as we lose the capacity to desertification, erosion, all really critically important. One of the big things that's big topic for discussion and research, particularly in Western Australia, is regenerative agriculture. Can we regenerate productive soils? Because we are losing them so fast. If my biggest concern about the long-term experiments, and I do check every so often, is that as soil science is not taught so much now in universities as a discipline, it's taught as part of other disciplines, that my old university in the UK is going to dig up those experiments just because we fought it for many years, but that soil destruction would obviously be an obstacle to continuing studies in this. But with these, as Andrew says, these long-term studies started out as agronomy trials, and then as different methodologies became available, so we were actually sampling them for a long time for actinomycetes that we were supplying to the drug companies looking for novel products. We then fatty acid data became available, and then molecular data became available, protein profiling, a whole pile of things. And we've been able to build up a very large database of studies on these soils. So it's really quite important to have long-term trials so we can do future studies. But let's hope we don't have global destruction of soils. So we have to protect them.
Yeah, sure.
Selma, how hard is the contributions of metabolomic studies in this report? I mean, how hard to do, or
Probably.
Yeah.
how hard to? When we started out doing this work, it was incredibly expensive, and everything was experimental. The methodologies were not proven. Now, if you've got the money, and it can be expensive, but the costs are coming down. Now it becomes relatively straightforward to do it. They're not difficult studies to do. A lot of our students are doing this type of work routinely. What worries me more is something Andrew alluded to, is that, you know, just having genomic sequences and just being able to show that this has changed based on 16S or 18S rRNA data isn't enough. We're losing the ability to do the process measurements, isotope modeling, isotope methods, tracing, things like that. Those are becoming much more difficult to do than the metabolomic studies. A lot of them are now largely automated. The slowest part is doing the extractions at the moment. The processing of the data is becoming easier. I run all of these analyses on my laptop.
Yeah.
Do you believe that is possibly change soil biochemical functions such as the rate of denitrification using different inoculated bacteria, or environmental factors have more important effects?
I think it's. I mean, I think you. I mean, the hope is that you can do that. Yes, and inoculum delivery becomes very important. And I think there is enough evidence that for that inoculation actually works. And you know, rhizobium, classic example. The important thing about the environmental factors is to make sure that the inoculant that you provide actually survives the inoculation, that it actually is not grazed out. Sometimes you don't want it. We did some work a long time looking at what happened to genetically engineered organisms added to soil. Fortunately, the protozoans ate them all, grazed them out. So that took away the risk associated with that. With denitrifiers, if there's a need for it, you might find that they find a niche and develop and grow in that niche.
Okay, thanks. I think that's all for questions. Thanks for your great talk. And
Okay. Thank you very much, Tony.
Andrew, have a good day.
Very nice
and
to see you.
Home soon.
Yeah, it's funny, we in this new online world, you're in Brazil right now. For us, you are anyway. So nice to have you here. Right
So we
now,
go to
six you and the three of us are sharing this with 60 other people. You've answered questions from people up in the Amazon, and you've been chatting to people from Embrapa, and you never even knew it. And also your friends here in Rio. And we look forward to seeing you here when you can come the next time when we can travel. In the meantime, stay safe, and we'll see you soon, I hope. Thank you very much, Tony.
Thank you. Thanks, Andrew. Thanks, everyone. Bye.
Bye. Thank you. Okay, I think we'll continue now, and I'll switch off my microphone and switch off my camera and pass over to Sulamita or Sheila, I don't know. And we go back into Portuguese. Bye for now.
Bom dia. Obrigada, professor Tony, pela excelente palestra. Agora nós vamos começar a passar os vídeos, as apresentações dos alunos da PBV que ficou para hoje. Então, a gente vai iniciar com o aluno Ricardo Esteves.
Só lembrando dos formulários, eu não sei se vocês estão conseguindo acessar, e a palavra-chave está na tela, tá? Qualquer dúvida, é só colocar no chat. E o primeiro vai ser o Ricardo, por conta de ele ter sido ontem, mas deu um probleminha técnico, nós vamos começar com ele. Depois segue o cronograma normal. Desculpa.
A próxima então será a Alana.
Pode ir lá, Sula.
Today I'm going to speak a little about my work entitled Development of Insecticidal Formulations Using Essential Oils from Jurubatiba Sandbank Species. Insecticides have been discovered in the 1940s to be used as pesticides in crops, but later they were weaponized and began to be used as biological weapons in wars and terrorist attacks. Here in Brazil, the Ministry of Agriculture began to show the registration of insecticides in 2005. In 10 years, 1,546 insecticides have been registered, and from 2016 to 2019, 1,605 insecticides have been registered. Since the change of government, the registrations of insecticides have increased at alarming levels, and most of those registrations are of moderate to highly toxic substances. Some of them are banned in Europe and USA. This data shows the importance of the research for new natural products with low or no toxicity for the environment. Our goal is to evaluate the insecticidal activity of essential oils and its nanoformulations from Restinga jirubatiba species. And to do that, we're going to collect species from the sandbank, extract their essential oils, perform the chemical characterization of the compounds, develop and characterize the nanoformulations of the essential oils, evaluate the insecticidal activity from the essential oils and their nanoformulations. The methodology chosen to extract the essential oil was the hydrodistillation with modified cleavager apparatus, and the chemical characterization was realized using a gas chromatographer coupled with mass spectrometer. The nanoemulsions were performed using the low energy input technique, and then topic assays were realized using the pure and diluted essential oil and with the nanoemulsions. Now I will present the preliminary result obtained with one of the species collected. A total of 99.05% of the essential oil was characterized. The major substances were beta-caryophyllene with 51.12%, alpha-humulene with 7.20%, and bicyclogermacrane with 4.83%. The topic assay with the essential oil and its dilutions showed a LC50 of 76.4 milligrams per milliliter and a LC90 of 135.9 milligrams per milliliter, demonstrating that this oil was active against the insect. And last but not least, the future steps are collect other species from the Lavacia family, extract its essential oils and obtain its chemical characterization, evaluate their insecticidal activity against several insects, evaluate their effects on metamorphosis and oviposition of these insects, compare their activities and develop the nanoformulation of the most effective essential oil, evaluate the LC50 and LC90 of the formulation, evaluate the ecotoxicity of the formulation.
Obrigada, Ricardo, pela excelente palestra, pela excelente apresentação. Então, dando continuidade, a próxima é a aluna Alana Almeida.
Hi everyone, I am Alana Pereira, PhD student from PBV, and now I'm going to present a little bit about my research that is entitled Bioremediation of Benzopyrene by a Fungal Consortium. First, I would like to introduce my advisors, Rodrigo Pires and Andrew McCray, and also the collaborators, Felipe Marbach from Federal University of Recôncavo da Bahia, and Eliezer Menezes from Federal Institute of Rio de Janeiro, Campus Pinheiral. The main objective of my research is to develop a formulation containing a fungal consortium for use in benzopyrene bioremediation process in contaminated environment. This bioformulation must be non-toxic, safe, cheap, and efficient in removing the contaminant. So here we have a big picture of the main methods. The research can be divided in 4 stages. The first one refers to sample collection, isolation, purification, and conservation of microorganisms and taxonomic identification. The second stage refers to selection of potential microorganisms, submerged fermentation, determination of enzyme activities, production of biosurfactants, and toxicity tests. The third one is about the formulation development that I hope to obtain a patent application here. And the last one is the evaluation of the formulation viability and the bioremediation capacity. Talking about my results, 56 fungal strands were isolated using a selective medium with rose bengal. Of these, 15 were selected according to their ability to degrade hydrocarbons using the redox indicator DCPIP. The microorganisms were evaluated according to their ability to produce lignolytic enzymes, lactase, manganese peroxidase, and lignin peroxidase. 5 strands were considered promising, especially the strand PIN270, that was the only one capable of producing the 3 enzymes. I am also working on taxonomic identification using both morphology and molecular techniques. 5 strands were identified at genus level, and we hope to find new species. To finish, I would like to highlight 2 important results. The first one is the submission of the manuscript from my qualifying examination. Revisions are now in process, and we await the editor response, and I hope it will be accepted by the end of the year. The other one is the patent application I was part of as inventor. So that's it. Thanks for your attention.
Obrigada, Alana. Parabéns, ótima apresentação. Sheila, seu microfone está desligado.
Falando a beça, né? Tá. Então, Alana, parabéns novamente. Todos elogiando que você falou bem calma. Então, a próxima apresentação vai ser da Bárbara Peckley. E nós temos ela teve um probleminha técnico na hora de gravar, então o nome vai sair com o nome de outra pessoa a gravação, tá? Mas o rostinho é da Bárbara e tudo certo. Pronto, Sula, se quiser, pode rodar.
Hi, I'm Barbara Pecco. I'm PhD student, and my work is entitled Development of an Agriculture Inoculant with Function of Biocontrol of Plagues and Bioremediation of 2,4 dichlorophenoxyactic acid used strain of agricultural soils. We know that Brazil is the second largest exporter of food. The model of production is chemical dependence. So Brazil is the largest consumer of pesticide in the world. In this pesticide, we have 2,4-D that stands out among the herbicides. It's the second most widely used herbicide in the country, and this substance can cause health problems and environmental damage. So the objective of this work is to produce biocontrol and bioremediation agriculture inoculant with bacterial strain from soil. My work has many steps. The first one is activation of the strain and identification. The second one is degradation of 2,4-D, that is the herbicide, by the bacterial strain. The third one is development of inoculant based on bacteria from soil. This part I produced the inoculant, and I did the viability of the strains during 1 year. The step 4, we studied the genomic ensemble and analysis. I group the cells, extract DNA, this had the sequence of genome of the strains, set up the genome with tools, Tringalori, we use Spades, we use Quest, Medusa, EDBA. So the results show me that the strain degrade the 2,4-D after 16 days, né? In minimum medium of 2,4-D. The strain one degraded, and the second one also degraded the 2,4-D. But bacillus thuringiensis have biotransformed the molecule of 2,4-D. These results I showed you last year. And the identification of the first strain is when I did the 16S rDNA, is closely related to Enterobacter, and the second one is Sucromobacter sinceri. So I showed you the identification and the viability of the inoculants. Show us that after 1 year, there are still live cells, Bacillus thuringiensis and Enterobacter. So we have 1 year viability and shows that the cells are alive, and it's possible to produce these inoculants. Search for degradation genes in the Has2 in the genomic parts. I upload the genomic of Cupriavidus pinantubonensis, that's similar Cupriavidus nectado, and the Hauschenotropha. It's a model of it's a strain model of degradation of 2,4-D. And I had Ochrobacter, Ciceria, and Enterobacter. When I compared in the host in the subsystem of metabolism of aromatic compounds, I can see the genes that are compared. And this study showed me that the strain Cupriavidus and Ochrobacter has the same genes in chloroaromatic degradation. 2,4-D is a chloroaromatic molecule. So in the part of chloroaromatic degradation, we have the genes, the same genes that suggest the same genes are involved in the 2,4-D degradation. I have to study more and explore more Enterobacter thuringiensis, and it's my my next step. So I want to thank you all. I want to thank you for all these years here, show my work for you.
Obrigada, Bárbara. Parabéns pela apresentação. E dando continuidade, o próximo vídeo é do aluno Cláudio Lopes.
Hello, my name is Claudio. I'm a student at the Biotechnological Bioprocess Plant Program, and I'm a doctorate student at the Federal University of Rio de Janeiro. And today, I would like to discuss my project with the public, and the title is Remediation of Metal-Contaminated Soils by Rhamnolipid Extract in a Liquid and Colloidal Gas Effluent State. My mentors are Lucy Seldin and Denise Freire. These are the laboratories involved and their activity along the process of our project, and all of them made significant contributions for the results that we presented here so far. So our research group is composed by Professor Lucy Seldin, Professor Denise Freire, Professor Ricardo Santelli, the postdoc Liv Araújo, the master degrees Fabio, Rui, me, Claudio, Francisco, Douglas, and Bernardo. So our objectives were to establish optimal parameters for heavy metal remediation from a short and long-term contamination site, evaluate the toxicity of the rhamnolipid towards soil bacteria after a heavy metal remediation step, a metal precipitation from the rhamnolipid extract, produce and characterize rhamnolipid CGA, and evaluate the heavy metal remediation as well. So, first off, we produce and characterize a rhamnolipid using glycerol, which is agroindustrial waste, and glycerol was the main carbon source for the production of a rhamnolipid in a bioprocess that was done in Labine. Afterwards, without any purification steps, we submitted the rhamnolipid to microscale remediation assay, where we tried to visualize with parameters such as pH, concentration, and time exposure, which was the optimal conditions for metal removal from the soil sample. And afterwards, we determined these heavy metals in soil samples, and also the concentration and conditions of the rhamnolipid were tested for toxicity, acute toxicity assays for commensal soil bacteria, and also to evaluate if it could do any harm to the environment. So this generated our first manuscript, and we demonstrated that it was able to remove a significant amount of arsenic, cadmium, and zinc from a short term, which is an artificially contaminated soil, and also a soil sample from a mining industry as well. Next off, we would like to applicate or use CGA, colloidal gas effluents. And why use colloidal gas effluents? Because of their better efficiency, which was demonstrated in the extraction of oil spilling, extractions of protein and oil in the food industry, and also bioremediation steps. So we figured that maybe we can also use CGAs for the remediation of soils contaminated with heavy metals. So we produced and characterized rhamnolipid CGAs and test the efficiency of these CGAs towards a soil contaminated with heavy metal as well. So a CGA is an afro is a colloidal structure, but a spherical structure, which is composed of a triple layered surfactant, a shell surfactant, which attracts air. So this unique conformation gives the colloidal gas afro stability and bubble diameters, which are very important in remediation steps that we'll discuss later. So we produce and characterize our CGA in a high-speed rotation agitator, and we measured the half-life of the CGAs, and also we visualize the average bubble diameter of the CGAs using microscope techniques. And therefore, this is the apparatus used in our remediation scale, and these were all the parameters that we established for visualizing stability and bubble diameter. So we did figure out that rotation and pH did play a role in the stability of the CGAs, and also bubble diameter, as well as time preparation. So we established a second experimentation where we tested these conditions in a microscale remediation step, and also the rhamnolipid extract in a liquid state. So this was the apparatus used for the remediation scale, and in a downflow or descendant flow rate, we we demonstrated that at this point, there was no significant remediation of the liquid rhamnolipid and the CGA rhamnolipid. Other parameters should be tested as well in order to better establish the CGA's efficiency in remediation, but the rhamnolipid extract did work in the macroscale remediation step. But now, I am in this point, the compilation and organization of my data and presentation of a doctorate thesis. Special thanks to the program and CNPq as well. And that's my presentation. Thank you all.
Obrigada, Cláudio. Então, Sheila, vamos dar um intervalinho de 10 minutos para o pessoal tomar uma água, um café.
Um cafezinho. Estou precisando.
Inventando palavras que eu adoro, precisado.
Enquanto está passando o intervalo, nós vamos deixar rodando o vídeo dos resumos gráficos, tá? Para vocês darem uma olhadinha.
Então, até daqui a pouco, pessoal.
Tchau.
Voltamos.
Olá.
Será que todo mundo já está de volta? Se puderem dar um alô no chat para gente, para saber se todo mundo já voltou do cafezinho, da água, para a gente dar continuidade às apresentações dos alunos. Então, vamos?
Pode começar. A próxima
começar?
aluna é a Juana.
Isso. Juana. Juana de Ramos Silva. Vamos lá, então.
Isso.
Hello, my name is Juana. I am a student of Professor Fernanda Abreu and Ana Maria Mazotto. The main goal of my work is to produce an enzymatic magnetic nanotube capable to degrading textile effluents containing azo dyes. At first moments, we created a soil microcosms and isolated bacteria tolerant to azo dyes. We selected bacteria capable of discoloring the azo dye metal orange with efficiency equal to or greater than 70%. And these microorganisms were tested in 4 other dyes in same group of azo dyes and are tested in the best strains were selected for the next steps of the experiments, which consists of discovering the enzymes involved in the decolorization process of these dyes, their mobilization in magnetosomes, and the effluent degradation tests and the toxicity of the final product of this decolorization. This project has a high chance of becoming a patent because similar research using enzymes immobilized in magnetosomes has not yet been found in the literature. So, in the preliminary results, 15 bacteria showed high potential of discolorization of the metal orange in static condition. Of these, 4 has a discoloration rate above 70% in all dyes except the gold yellow dye. These microorganisms were initially identified by Maltstoff, and three strains are bacilli and one is Pseudomonas. Our perspective are to find more bacteria capable of degrading these dyes and to find lacases, aziridatases, and polyphenol oxidases as enzymes involved in the decoloration of azo dyes and immobilize them in magnetosomes, maintaining the potential of discoloration. We also expect that the final product after enzymatic treatment be non-toxic and completely discolored. Our next steps consist in finalize the selection step, find out which enzymes are involved in the coloration process, and immobilizing them in magnetosomes, and carry out the coloration and detoxification tests with this nanotube. Thanks, guys, and it's all.
Obrigada, Ruana. Agora nós vamos passar o vídeo da Helena Oliveira.
Hello, my name is Helena, and I'm going to present a project entitled Analysis of the Factors Influencing the Dominance of Hydrogenotrophic Methanogenesis in Anaerobic Environments. During the anaerobic degradation of organic matter, there is the production of acetate and hydrogen and carbon dioxide, which are the two main substrates for methanogenesis. Although the reaction suggests that 70% of methane should be produced by acetoclastic methanogenesis, the hydrogenotrophic pathway is often predominant. The factors that cause this pathway shift are not yet clear, but there are some indications such as high temperature, low biodegradability, low pH, and high concentration of acetate, hydrogen, and ammonia nitrogen, which favor the hydrogenotrophic pathway. The aim of this study is then to determine the factors that influence the metabolic pathway of methane generation in anaerobic environments. In order to achieve that, a literature meta-analysis is being currently conducted, and in the future, experiments to evaluate the influence of the main factors will be carried out, with the pathway being determined by stable carbon isotope analysis. I'm going to present the preliminary data from the meta-analysis. In this graph, we can see the influence of the substrate type to the amount of hydrogenotrophic methanogens in the reactors. The blue scale represents the contribution of hydrogenotrophic methanogens, with darker shades corresponding to a higher contribution. Higher hydrogenotrophic content was found for reactors operating with municipal wastewater, food waste, and agricultural residues. In terms of the applied temperature, we can see that the higher the temperature, the higher the contribution of the hydrogenotrophic pathway. It is the dominant pathway in 62% of the data with thermophilic reactors, while it dominates only 42 and 34% of mesophilic and psychrophilic reactors. Looking at influent pH in continuous reactors, we can see a trend. Excluding the lowest pH values, which only had 3 data points and therefore cannot be representative, we see that for both high and low pH values, the contribution of hydrogenotrophic methanogenesis is elevated, with acetoclastic being the main pathway in more neutral pH values. For the total ammonia in batch reactors, apparently a higher contribution of the hydrogenotrophic pathway is perceived when ammonia concentrations are higher, from 2,500 to 3,500 milligrams per liter. However, when looking at continuous operation, no trend can be observed, indicating that maybe in this case, ammonia and nitrogen alone would not be a decisive factor for the methanogenic pathway. On the other hand, if we look at the carbon-nitrogen ratio, we can again see a tendency. The lower the CN ratio of the substrate, the higher the contribution of hydrogenotrophic methanogens. And a low CN ratio indicates a higher nitrogen content in the influent. This could indicate that the relation between ammonia and carbon is more important than the ammonia content alone for pathway dominance. In the future, there is a need to evaluate if the observed trends remain after all data is collected, and to compare the methods of pathway evaluation, such as microbial analysis and stable carbon isotope analysis. Also, we will perform a multivariate analysis in order to observe the influence of combined factors in the methanogenic pathway. And finally, the conduction of experiments to evaluate the trends pointed out by the meta-analysis. Thank you for your attention.
Obrigada, Helena. A próxima agora é a Érica Santoro. Vai lá, Eriquinha. Princess do Mar. Vamos lá, então.
Hello, everyone. My name is Erica Santoro. I'm going to present my PhD project here, which is entitled as Microbiome of Thermal-Resistant Corals, and also the manipulation of beneficial microorganisms for corals and Musselsmelia ispida. This intend to cope with thermal stress. So my supervisors are Raquel Peixoto and Andrew McCrae. And my PhD is divided in 2 chapters, which first of all, I could produce this paper where we submitted to ISNE Journal, which is it's called Opening the Heat Box. Multi-domain analysis reveals different functional and microbial association with thermal-resistant and sensitive corals. And the main results that you could observe was that we found resistant and sensitive corals in the field in Bahia. So we caught these colonies and did some experiments, thermal experiments with these colonies, which we could see different in the physiology and the microbiome of these corals. So here we can see this is a photosynthetic efficiency of the coral, where the sensitive was like decreasing during the experiment, during the heat stress, and also changing a lot the microbiome. And on the other hand, the resistant corals did not change that much, and they could keep the photosynthetic efficiency during the heat stress, as well as did not change that much the microbiome. So this was the main results of this paper, where you could see that microbiome may influence the thermal resistance of these corals. Okay, so after that, there is this second chapter of my dissertation, which calls Microbiome Manipulation Elicits Metabolomic and Genetic Restructuring to Mitigate Heat Stress and Evade Mortality, where we produce a probiotic with beneficial microorganisms for corals and use that probiotic to protect and mitigate the heat stress and the mortality in corals. So this paper we just submitted this paper in the Nature Communication at this month in December 2020. And the main result, the main findings we had was that we could see that the manipulation of BMC, of the beneficial microorganisms for corals, increased in 40% the coral survivorship, as well as it elicited the host metabolic change. And when I'm talking about the metabolomic, I'm talking about DMSP, DMSO, and lipids, which are really important metabolics for corals. And they also downregulate the manipulation of the BMC, also downregulate apoptosis inflammatory system, which maybe are involved with the increase of survivorship. And also the BMC elicit upregulation of genes for oxidative and thermal stress control. So these are the main results of this paper. And this is my academic production and experiences in the during my PhD, during this process, during these 4 years. As I told to you guys, I'm defending in few days, in December 11th. And after that, my next steps is going to Caus in Saudi Arabia to do my postdoc. So this was one of the experiments I have during my PhD, was going to Biosphere 2 in Arizona, that I could get this scholarship from CAPS Print. And also I had the opportunity to go to HMB in Hawaii, where I was developing part of my PhD as well, using probiotics to trying to save and protect the corals there in Hawaii. And that is it. That was a resume of my PhD project, and I would like to thank everyone from my lab mates, my supervisors, and all the collaborators that was part of this. Thank you, everyone.
Parabéns, Érica. Amanhã a Érica defende a tese de doutorado dela.
Parabéns, Érica, e boa sorte amanhã para você.
Sim,
vai dar tudo certo.
Vai arrasar.
Com certeza.
Sheila, com as apresentações?
Isso, continua. Agora é o João Barreto.
Isso. João tem uma pesquisa maravilhosa, todas são maravilhosas, mas a do João é super inovadora.
João é especial.
Sim, principalmente para nós mulheres. Também alguns homens que gostam de batom. Então vamos assistir a dele.
Hi everyone, I'm João Barreto, and the advisor of my project is Alane Beatriz Vermelho. I'm a PhD student, and the title of my work is Microbial Pigments Applied to Industry. The general aim of my job is developing cosmetics from microbial pigments, and the methodologies are going from bioprospecting and isolation of the microorganism to manufacturing a prototype line of those lipsticks. But why are you doing this? Because of the environment, because today the traditional synthetic dyes that are used for the formulation of cosmetics are not biodegradable, and we have some natural choices like mineral dyes, but they're not good because they are not biodegradable as well, and they can be contaminated with some heavy metals. And the vegetable dyes are not that good either, because the plantation of those vegetables just to extract their pigments takes time, pesticides, and creating competition for arable lands. Instead, we have a microbial choice that can be produced in bioreactors. We can control all the physical and chemical conditions of the process, and we can optimize the whole process using experimental designs and some genetics modification. And here in this table, we have some other advantages of this microbial choice. Those benefits are listed here, and we have some like biodegradability, it's free of contamination, is a renewable source, we can have some substrate utilization, water economy, it's weather resistant, and so much more. So now we have two pigments from two different microbes that are hyperpigmented. We have optimized the medium with carbon and nitrogen sources, pH, temperature, instance of light, and oxygenation. We have also uh, established all the extraction with organic solvents. We have those. We have also best formulation of the lipstick with those pigments, and we have quantification with UV light. Other tests that we did are cytotoxicity tests in macrophages, fibroblasts, and keratinocytes. And we can see here that not only pigment A, but also pigment B have a great result with a good cell viability. And we have also the DPPH test, and it measured the antioxidant potential, and they're great too. This can contribute to save money from the formulation because with a great potential and great antioxidant potential, we can save money with we don't have the need to use another antioxidant from other sources in the formulation. And we also, in this year, we had a patent application of our formulation. Its application is in INPI, and these are the number, and these are the photo of my very first lipstick. And the next steps are the characterization of those molecules, the production bioreactor, the test of sun protection factor, and produce a prototype line of these lipsticks and test their stability to heat, solubility, color duration, and estimation shelf life. And finally, write and publish an article. Thank you, everyone.
Obrigada, João. Parabéns pelo trabalho, pela apresentação. Não vejo a hora de comprar seu batom nas farmácias. Bom, então vamos seguir com o próximo aluno, que é o aluno Marcelo Fernandes.
Hello, my name is Marcelo, and I am a master student of PBV. My project is about the communication between grass crops and diazotrophic bacteria and how to identify a beneficial association between them. My advisor is Dr. Adriana Emerson, and I'm part of the LBMP lab. The main goal of this project is to find bacteriogenetic markers of the benefic associations with grass crops, like sugarcane or maize. These markers can be anything from a gene to a full pathway or structure that is over or underexpressed when the benefic association occurs. And by that I mean when the plant expresses a characteristic of interest for us after the inoculation process, like increased dry mass by sugarcane, for example. For that I have two approaches. An inoculation experiment using Arabidopsis thaliana as my plant model, and a list of Azospirillum brasiliensis as the inoculant agent. And in this step, I'll be using a pretty recent mixed method of DNA quantification for bacteria detection that I'll be talking about in a second. And the other approach will be a series of bioinformatic analysis, not only for my experiment, but also on some transcriptome data from previous inoculation experiments from the lab I'm in, where I'll be also looking for these markers in different species. What we expect to find from this experiment, the switch of the mutants we'll be testing can cause beneficial responses in the host after the inoculation process. And for that, we'll analyze both the phenotype of the host and the colonization efficiency of the bacteria. which where we will use a mixed method of absolute DNA quantification, carrying the specificity of the digital droplet PCR with the accessibility of a qPCR, real-time PCR, resulting in a precise and replicable method of bacterial detection in inoculation experiments. We will use bioinformatic analysis to better understand and describe any of the effects we observe, and also try to link these results with the transcriptome data results we already have. And as next steps, we expect to be able to predict, at least partially, the responses we will get between a specific host and bacteria without needing a full inoculation field test, with both time and knowing consuming. We also expect to establish a new way of bacterial detection that is both precise and replicable for inoculation experiments, and for a more distant future, we may be opening a path for new inoculant formulas, either by using these azospirillum mutants we'll be testing, or by exploring biotechnologically these genetic markers we'll be finding, increasing the efficiency of the existing inoculant formulas. And that's it. Thank you.
Obrigada, Marcelo. Parabéns. A nossa próxima apresentação é da Maria Chacon, a nossa aluna venezuelana.
Hi everyone, my name is Maria Alejandra Chacon. I am a master's student at the Plant Biotechnology graduation program at the Federal University of Rio de Janeiro, and today I'm going to present to you my master's program, my master's project, which is entitled The Utilization of the Coral on a Chip Model to Optimize the Coral Probiotics Approach to Improve Coral's Health and Growth. My tutor is Raquel Peixoto, and my co-tutor is Elena Vilera. Here in this section, I'm going to show you a little bit about the specific objective of the thesis, as well as the methodology we're going to use for accomplish our objectives. So first of all, we're going to take the coral Poecilophora armicornis, and we're going to increase the salinity. When we increase the salinity, the coral is going to expel a little polyp, and that little polyp is called a micropropagate. Then we're going to observe which corals best adapt to the different stressors in which they're going to be exposed. We take these corals and we select a consortium of beneficial microorganisms for the corals. We take these BMCs and then we inoculate the new micropropagates, and then we're going to observe how they react or how they evolve using the coral on a chip model. So for the observation, we're going to insert these new micropropagates of Poecilophora armicornis that have the BMCs, and we're going to observe them in the coral on a chip model. This model is actually a very cool model because it allowed us to observe in vivo experiments. After that, we're not only going to observe how are the interactions between the BMCs and the coral, but also we're going to observe the calcification and the growth of the coral. For this, we're going to use high magnification microscopy, and we can see the daily growth of the coral, observing the rings of the new deposit calcein. With this model, we are going to be able to observe the coral not only as an organism, but also as a meta-organism. So with this, we can see the coral's interactions with the microorganisms as well as the symbiosis between the coral and the zooxanthellae. And also we can see how different BMCs of different corals that have been exposed to different treatments can impact the health of these new micropropagates. Finally, we're going to use electron microscopy, and we're going to employ fluorescent dicalcine so we can observe the pattern of calcium carbonate deposit in the settled micropropagate. So before talking about the expected results, I want to talk a little bit about why I wanted to do this project. First of all, many species depend on coral reefs. Also, a percentage of the oxygen of the earth comes from coral reefs. That's why we want to develop new techniques that could improve the coral resistance and resilience to different anthropogenic impacts. This could actually cause the metaorganism's dysbiosis and eventually the death. As I mentioned before, one of the advantages of the coral on a chip model is that it allows us to observe in vivo results or experiments. This is a huge advantage because there's a lot of variables present in the ocean as well as in the aquarium systems. That's why it has been a challenge to study the beneficial microorganisms community that is associated with corals. So, as an expected result, it's the creation of a microfluidic platform. In this platform, we want to observe not only the micropropagate, but also the beneficial microorganisms for the corals. We want to observe the calcification process as well as the interaction between the coral and the zooxanthella, as well as between the coral and the microorganisms that surround it. Here we have two examples. Another expected result is the consortium of beneficial microorganisms for corals. In this experiment, we're looking for very specific things that we want that our beneficial microorganisms for corals consortium have. That includes nitrogen fixation bacteria, as well as carbon cycling and regulation. We want to observe the photosynthesis and how the zooxanthella can improve it. We want to observe the production of DMSP as well as the degradation of the TMSP. We want to observe the production of quorum sensing in the bacteria, as well as the production of microsporin-like amino acids. And we also want to observe if the bacteria that we are collecting have bacteriocins. So for the conclusions, we have that this microfluidics platform will allow us to take a more precise look into the effects of the BMC's manipulation, including their mechanisms of interaction with the host and how they can improve the fitness and the growth of the coral. Thank you very much, and I hope you enjoyed my presentation.
Obrigada, Maria. Parabéns. Agora vamos passar para o próximo aluno, que é o aluno Matheus Uchoa de Oliveira.
Hello and welcome to my project presentation. My name is Matheus Oliveira, and our project is named Antimicrobial Potential of Actinobacteria against Fecal Pathogens. I'm advised by Professor Fernanda and co-advised by Professor Rodrigo in association with laboratories mentioned below, LabM and LabMax. Through our objective, we can basically say that through bioprospecting, isolation, and testing of new actinobacteria strains already obtained from different Brazilian environments based on their biocontrolling of phytopathogenic agents like fungi and bacteria, it's possible to develop a bioformulation capable of being used in functional and optimal way in agricultural practices, following the steps below. We can see here that starts on bioprospecting through isolation and then some antibiosis testing, so we can see the biocontrolling effect. Then through electronic microscopy, then genetic identification, fermentation, and then the development of formulation and its viability testing in greenhouses with 3 specific plants: banana, papaya, and corn, and finally achieving to our final goal. But we have a problem. The problem is that Brazil has about 40% of the country's soil area occupied by agricultural establishments regarding the Sahara and IBG. And obviously, one of the biggest problems is regarding the sustainability in the presence of phytopathogens of these crops, and also the abusive release of new pesticides, which ends up eliminating the natural biological controlling fauna, and also a tremendous damage to the environment and the health of its consumers in the final steps. We can exemplify the Fusarium cisperan to the left, which is attacking a banana tree, and Xanthomonas campestris attacking a lettuce, which also attacks many other kinds of crops. Through the project, we have some expected results, of course. One of them is the isolation of a reasonable number of actinobacteria and test them for their antimicrobial potential, which obtaining a strain capable of inhibiting or destroying an its own pathogen, its biocontrolling effect, of course. Subsequently, there is the evaluation of the interaction degree between the selected microorganism, the actinobacteria I mentioned, and the phytopathogens tested. And after it, then identifying them on a molecular level. As future steps, we can mention the enzymatic assays for a better quantification of such potential, together with the development of bioformulation, focusing on an easy spreading effect, together with integration of the environment, and, if possible, a familiar form to consumers of these projects, focusing on the economical. And also a viability testing in controlling environments, as I talked before, in greenhouses following the 3 plants I mentioned too: banana, papaya, and corn. We have also to discuss some points, some advantageous points. One of them is that actinobacteria are true metabolic biofactories. They generate a huge spectrum of biomolecules, from enzymes to biofactories and many others, proteins and many other inputs, which make us think the development of a formulation based on a biocontrolling actinobacteria in Brazilian crops without causing collateral damage would be a strategy of great interest, not only for us, but for many other countries outside of our circle. And of course, the main point is that a formulation with microorganisms from Brazil's own territory, reducing import costs of these products, would be an extremely advantageous economically and environmentally speaking, since external microorganisms would not be inserted as a part of our ecosystem, changing the natural order of the city. Well, to all present here, I thank you for your time and
Sheila, cadê Sheila?
Oi.
Vamos dar uma pausa, passar os resumos gráficos agora?
Isso, agora já acabaram as apresentações dos alunos da parte da manhã. Então, nós vamos rodar mais uma vez o resumo gráfico que estão pedindo. E depois do resumo gráfico, nós vamos entrar para o intervalo do almoço. Lembrando que 1h nós temos a palestra com o professor Andrew, falando sobre Cap Sprints. Então, é muito importante para quem quer fazer doutorado de sanduíche. Enfim, muito bom e vamos assistir.
Sim. Está indo muito rápido, né? As apresentações.
Muito.
Sim, sim.
Todo mundo muito organizado.
Sim, o nosso cronograma está adiantado.
Mas eles estão de alto nível, hein? A apresentação. A apresentação aqui de alto nível.
Sim, com certeza.
Ao vivo presencial não fica assim, né?
Não, é muita correria, não tem como.
A correria, apresentando em cima de um palco com microfone. Acho que aqui tá mais tranquilo. Então,
mas tá todo mundo sentindo falta, fala aí.
Sim, estamos todos. O pessoal está comentando no chat.
Todo
também
mundo
no nosso
falta do churrasco, né?
Principalmente.
Hoje teríamos o nosso churrasco vegetariano, churrasco para não vegetariano.
Sim, com certeza.
Com cerveja artesanal, fabricada
Cadê
pelo
Zé Roberto?
Pelo Roberto, professor Roberto, isso aí.
Mas não tem problema. Depois da pandemia, nós vamos nos reunir para fazer o nosso churrasco.
Sim,
Sem problemas.
Vamos esperar. Esse momento está sendo difícil para todos nós.
Isso.
Então, o Andrew comentou aqui, churrasco, quero. Queremos, Andrew. Então tá, Tarcísio, você vai rodar os gráficos pra gente, então?
Sim, liguei, vai rodar os gráficos.
Então tá.
Tá ótimo, então.
Acabar o resumo gráfico, a gente volta para dar um intervalinho, então, para o almoço.
Tá ótimo. Até daqui a pouco.
Certíssimo. Agora nós vamos dar um intervalinho para vocês. Vai rodar mais alguma coisa? Vou deixar a palavra-chave, né? Nós vamos dar um intervalo para o almoço e vamos voltar 1 hora com a palestra do professor Andrew.
Sim,
Vocês querem falar mais alguma coisa, comissão?
Não, é isso mesmo. Perfeito.
Não.
Então até daqui a pouco.
Até.
Sim, nos vemos 1 hora. Bom almoço, pessoal. Até lá. Até já.
E com alguma surpresa, hein? Estamos ansiosos.
Ah, é? Tem surpresa?
Tem na apresentação do Andrew, ele já falou aí no chat.
Então tá. Tô aqui ansiosa, professor Andrew. Espero que seja um sorteio.
Não sei.
Um sorteio, um brinde. Essa semana já ganhei um brinde, hein?
É.
De apresentação. Então tá. Até mais.
Tá
pessoal.
ótimo. Tchau.